Now we will look at some really interesting experiments on the neurochemistry of hope, also involving the use of placebos. And these were experiments were attributed to a man named Benedetti, who in fact wrote an entire book on placebos. So as I said last time, the notion of placebos has taken on a veneer of scientific legitimacy, has become the subject of intense exploration, and now is something that seems to have the capacity to legitimately establish hope as a real medical phenomenon. This module is coming to you in 3 parts. First, we're going to talk about the basic experimental setup used by Benedetti. Then we're going to talk about some variations on a theme and a particularly interesting challenge to the idea of hope as a medical phenomenon in part 2. And then lastly, we are going to move out of the realm of pain intervention and talk about placebos and hope in another venue where it's truly extraordinary. So let's get started. So we're going to be taking on hope as a medicinal intervention. And you recall that in setting this premise up, we talked a bit about it in the last module. We noted, for instance, that we have natural opiates in our brains doing their thing. They include the endorphins and enkephalins, and that these materials, among other reasons, are released by having some sort of belief and expectation of a particular outcome. And these things, belief and expectation, are the ingredients that we can identify most readily as being the basis of hope. And so, medicinally, what seems to be going on is that the release of endorphins and enkephalins basically mimic what morphine does, in that it can cause pain suppression. And this is the basis for the experiments conducted by Fabrizio Benedetti of the neuroscience department at the University of Turin in Italy. So this is the basic way in which these experiments and the variations upon the themes were conducted. They began with somebody who was identified as a research assistant, also abbreviated RA. And so this guy, the research assistant, was dressed to the nines in an attempt to make him look like a real medical person. He was not a doctor, nor did he play one on TV, but he was dressed like one would be dressed if you went into a regular hospital or clinic. You would expect to see somebody dressed pretty much like this guy. So he was wearing a crisp, clean, white lab coat that gave the illusion of being a proper physician. It probably had a stethoscope and so forth to increase the deception. We also have a person who is basically the test subject, called the volunteer, sometimes referred to as the V. And the research assistant, remember, the doctor-looking person, would say in a very authoritative basso profundo voice, "I'm going to put a cuff on your arm and tighten it until it hurts." And so the RA would wrap the cuff around the arm of the volunteer, tighten it up, and wait until the volunteer showed evidence of pain, like wincing, "Ow, that hurts." And it should be noted that the volunteer, in addition to having this cuff on his arm, was also attached to monitors, which were able to record and measure physiological reactions to the painful stimulus, which, of course, was the tightening of the cuff. And so the monitors would pick up increases in heartbeat that came from tightening the cuff, sweating, blood pressure, and muscle contraction, all possible reactions to a painful stimulus, which again, in this case, was tightening the cuff. So all of this gets recorded, and then once that was done, the RA would release the pressure and measure the decay of those physiological indicators of pain as the pain faded away. So that's the basic experimental setup. Two guys, one of whom is the medical doctor appearing person, or RA, the second of whom was the research recruit or volunteer who subjected himself to the things that were done in the experiment. Okay, so that was the basic setup, but then we have some variations on that theme. So having already measured the level of tightening that it took to induce the painful response, in the next round of the experiment, the RA would say, again in his authoritative sounding voice, and prior to tightening the cuff, he would say, "This time I will give you morphine to prevent the pain that you felt previously." So he would then bring out a syringe, visibly fill it with morphine, and inject it into a portal that had been established in the arm of the volunteer. So now the volunteer has morphine coursing through his system, and the cuff is again tightened to the level that previously caused pain, but this time there's no indication of pain. The volunteer doesn't wince, and there are no physiological indicators of pain on the monitors. So we can conclude from this, not surprisingly, that morphine prevents pain, as we've been discussing already. You know what the mechanism for morphine preventing that pain, namely the morphine binds to the on cells in the spinal column. It prevents the on cells from doing their thing, but gives the off cells the ability to act as circuit breakers, and that prevents the pain. Okay, so here it is for you, spelled out step by step. The morphine blocks the on cells, allowing the off cells to do their circuit breaker thing, and as a consequence, when the volunteer has morphine in his system or her system, the signal doesn't make it to the brain, and the volunteer feels blessedly no pain whatsoever. But we're not done yet. Oh no. There was a third experiment in the sequence. After the previous bit with the morphine was done several times so that the person kind of got used to seeing the morphine being injected into the portal and then experiencing no pain after that was done several times. Next, the research associate told the volunteer that he would again be injected with morphine, as he had been at this point several times before. But this time, they played a little trick on the volunteer. Unbeknownst to the volunteer, the injection that he got was not morphine, but rather an innocuous dose of saline, basically of water, that had no pain-killing ability whatsoever. But the volunteer was still under the impression that he was receiving an injection of morphine. So the cuff, again, was inflated, recalling now that he has not morphine but saline in his system. What should happen? Well, if you said it should hurt, you might be correct, because he doesn't have morphine in his system, right? It's just water. It has no pain-killing ability whatsoever, and so he should feel pain, right? But no, that is not the evidence given either by his wincing, which didn't happen, or by the physiological monitors of pain on the screen. There was no evidence whatsoever of pain on the part of the volunteer, even though he or she received no pain-killing morphine this time, but rather saline. So what's going on? Yep, the volunteer showed no sign whatsoever of discomfort, no wincing, no signs of physiological pain or stress on the monitors. So in essence, what happened experimentally was that instead of receiving the promised morphine, as he had been promised and received several times before, this time the little trick was played and a placebo or innocuous saline was injected into the portal. But here's the interesting thing. It had the same effect as morphine had previously had on the volunteer. Now that is interesting. So here's what Benedetti thought and his hypothesis about why this was going on. The use of saline in, I guess we should call it the third experiment because that's how I addressed it previously. In the third experiment, did not shut off the on cells. It had no capacity to shut off the on cells, right? Saline doesn't have any neurological activity whatsoever. As a consequence, when the volunteer got not morphine but saline, the off cells should not have been able to exert their circuit breaker influence, which suppresses the pain. So how do we explain this? Well, according to Benedetti, the hope and expectation that had been previously established by actually getting the morphine, this time the hope and expectation caused the release of natural opioids, which we had previously characterized as endorphins or enkephalins, from the brain, which did have the capacity to turn off the on cells and allow the off cells to shut down the pain. Okay? So in this case, morphine is doing nothing because there's no morphine in the volunteer's system. But the expectation that morphine would be provided resulted in the release of endorphins, which are natural opiates and did have the capacity to bind to the on cells, shut them down, and allow the off cells to exert their circuit breaker activity. Now, to test for the legitimacy of that interpretation, Benedetti conducted yet another experiment. So this time, we have another sleight of hand experiment. The volunteer is going to be told one thing and then another thing will be done to him. But the point of this experiment is to confirm that what is happening is that endorphins are being released when the volunteer expects to receive morphine but does not, instead receives saline. And so now Benedetti wants to confirm via another means that that is in fact what's going on. So again, the RA tells the volunteer that he's going to experience this painful stimulus, and indeed, when the cuff is tightened and there's no chemical intervention, then the volunteer shows the expected painful response. He winces, and there are physiological indicators of pain on the monitors. Again, we go to the step of the RA telling the volunteer that he will get some morphine to block the pain before he tightens the cuff again. Now, when morphine is given, the pain is in fact blocked. So again, that is something we've seen before. That's not terribly surprising. But next, the RA fills the syringe not with morphine or with saline, but with a drug called naloxone. Naloxone is a drug that specifically blocks the receptors on the on cells and prevents endorphins from binding to those on cells. So you see where we're going? We're going to run another experiment in which the volunteer is told he will be given morphine, but is in fact not being given morphine. If it were saline in the syringe, we would expect to have the release of endorphins that proceed to bind to the on cells, shut them down, and allow the off cells to have their circuit breaker activity. But in this experiment, we're adding not saline to the syringe, but this other drug called naloxone. And this drug specifically binds to on cells, but in such a way that neither morphine nor endorphins, if they're released, can bind. And so this experiment should allow us to determine if, in the previous experiment, it was in fact endorphins shutting down the pain. So, Naloxone is now in the syringe. It goes into the volunteer system, and we know that its mode of action is to bind to the surface of the on cells. And it does so in such a way that when endorphins are released by the expectation that there's morphine in the syringe, they'll be released, but now they can't bind. So they can't have their activity of shutting down the on cells and allowing the off cells to do their thing. Because the volunteer is set up to feel hopeful that he's and expecting to receive morphine, he will go ahead and release endorphins as before. But now they can't bind to the on cells, and therefore they can't have their effect. The volunteer still thinks he's getting morphine, but he's actually getting a drug to prevent the endogenously released endorphins to do their thing. The endorphins, of course, are released because a volunteer has been primed to have the hope and expectation of no pain, and because he was told that the syringe that is going into his portal has morphine in it. But even though endorphins are released because of the previous set of expectations, the naloxone in that syringe overrides the binding of the endorphins, even though they're released. The off cells can't break the circuit, and the volunteer does feel pain. He winces, and the physiological monitors show evidence of a pain response on the monitors. So, at this point, can we then conclude and suggest that this thing we call hope really is just endorphins in your system? And I think it's fair to say that yes, that's true, at least in part, because that is the biological and neurochemical system we have. The expectation of a pleasant event causes the release of endorphins, and at least in the realm of pain suppression, where we know what the mechanism is and we can manipulate its response, yes, there is an equation of hope with endorphins, at least in part, because belief, expectation, and desire can cause the release of those endorphins. They are natural opiates, and they do have a role in pain suppression. We can further stipulate that if nothing interferes with those endorphins, like naloxone, for instance, then the volunteer should feel no pain, even if there is a painful stimulus present. If the receptor to which the opiates bind to produce their effect is blocked, such as when naloxone is administered in the syringe, then the pain suppression effect will not be realized even though endorphins are released. So, yes, endorphins and hope are in a sense synonymous. Another way to look at this, though, is that a change in mindset can reinforce the effectiveness of a belief in a particular medicine. All right, so at this point you might say, hey, well, that's pretty cool, yeah, and in the realm of pain suppression or pain relief, yeah, you can have this internal biochemistry, the mechanism for which is known or at least understood in terms of preventing pain. Cool. But is that it? Is that the scope of hope in humans, the possible elimination or reduction in pain when painful stimuluses are present? And the answer is, well, we don't really know for sure, but we do have some other experimental evidence that suggests that the capacity of hope to produce a salutary benefit in medical terms may extend beyond the somewhat minor examples we've looked at thus far. And this is a really interesting study for a couple of reasons. One is that it goes well beyond minor pain suppression. It also suggests that the power of the mind in determining medical outcomes may be quite a bit broader than we originally thought. So let's take a look at this as something that might prove to be instructive as we go along and as medicine comes to grips with what's going on in the brain with respect to the outcomes of medical interventions. This particular study has to do with the condition known as degenerative arthritis, which, if you've had it, even in a small amount, is very painful and is incredibly debilitating. As the name suggests, it's a condition that takes place in the joints. And when anti-inflammatory drugs that can be given to correct this fail, surgery can be the only option left. In this particular study, the surgery studied was performed on the knees of the individuals who were affected. The surgery is performed through an arthroscope so that the surgeon can see the inflamed and degenerating cartilage, and what they do is to shave it down and then flush out the inflammatory cells so that the joint can heal. It's a fairly common procedure, over 500,000 procedures per year. The cost at the time that I wrote this was about $5,000. It's probably way more than that now. So it's both common and fairly expensive. If we had some other way of dealing with this, it would be a good thing. Now, the article I'm talking about was published in 2002 in the New England Journal of Medicine, which is one of the premier medical journals in the United States. This is not a fly-by-night or questionable quality journal. This is one of the gold standard journals. It's a serious journal. So the fact that this particular article was published in that specific journal is remarkable. And what they did, to make a long story short, is that they demonstrated the positive impact of placebos on pain of arthritis in the joints. Now, you saw the picture previously of what they do to fix it surgically, and we talked a bit about the nature of what's going on in the joints. The cartilage is breaking down and the cells are inflamed. So this is a really interesting finding, right? Let's talk a little bit about the specifics of the study. The investigators rigorously assessed the levels of pain before and after surgery. But what they had done to conduct the study is they divided their patient population into two groups. One received the real surgery, the actual repair and correction of the bad cartilage in the joints. But the other group received a sham operation or a placebo operation. They didn't fix the problem. They had 180 patients total, and the patients were randomly assigned to either treatment group. And the surgery was conducted on the patients in the same way, regardless of which group they belonged to, in the sense that the patients in the placebo group were prepared in the same way. You know, they were given the pre-operative suite of procedures. They had their knees washed in the same way. They were taken into the operating room. They were given the pre-operation anesthetic that kind of makes you woozy. They were taken into the operating room. They were put under full anesthesia. A small incision was made in the knee for both groups. but only the surgical group got the actual surgery, in which they scraped out the bad cartilage and flushed out the inflammatory cells. The placebo group did not receive that intervention. They just got the hole made in their knee sewn up, and then patients in both groups were put into the post-operative suite in the same way. They were allowed to recover for the same amount of time, and they were treated exactly the same after the surgery was done with respect to things like rehabilitation, pain management, and so forth. So what were the results in these two treatment groups? Well, post-operatively, as I said, patients in both groups got the same treatment in terms of painkillers, walking aids, physical therapy, and so forth. Furthermore, both groups of patients were followed for a full 2 years after the surgery had taken place. During this time, they were assessed at various junctures for levels of pain and changes in functionality and ability as a result of their surgical experience. So one would expect that those individuals who'd had their joints actually cleaned out and the inflammatory cells removed would do demonstrably better, right? Because this is a physical problem. And the people who actually had the real surgery had their physical problem fixed. In contrast, the people who had the sham surgery did not have their physical problem fixed. And because the nasty degenerating cartilage and the painful inflammatory cells are still there, you'd expect for them to also, not for also, but you would expect for them to continue to show the symptoms of arthritis. But here's the thing. Both groups did equally well. The placebo group showed an equal benefit without having had the actual surgery. This is crazy. Wow. So how could this happen? As I said, arthritis is a physical problem. We know what causes it. We know how to fix it. The knee gets clogged up with that nasty cartilage and those inflammatory cells. The cartilage needs to be removed. That's how we fix it. And the inflammatory cells need to be flushed out. It's pretty simple. But in this experiment, apparently, the belief and the expectation that the individuals who really got the placebo treatment led to the expectation that, yeah, I got the real treatment, and that expectation released those powerful endorphins that blocked pain. But they blocked pain over a 2-year period. Is that even possible? And this goes some way to answering the question that Bruce Cohen posed in the previous module about what is the duration of a hope-induced effect? Well, in this case, it looks like 2 years. And that was the length of the post-operative study period. How do we explain this? What we found among the people who got the placebo treatment was that both their reduction in pain and their increase in functionality was the same as the treatment group. So one hypothesis about how this is possible is that the pain that the placebo group was experiencing prior to the sham surgery was an impediment to exercise. And once they had the placebo-induced release of endorphins, there was enough pain suppression that they could exercise productively, and the exercise led to an increase in functionality. This allowed for their rehabilitation to proceed in roughly the same way as the actual treatment group, and so that could explain, or at least help explain, why the ability to use the joint, which had not been cleaned out, more productively, even though it had not received the corrective surgery. And so, yeah, we can make a lot out of this study, and maybe it's just a good down payment on something that needs to be followed up with further studies. But it does raise the interesting and hopeful possibility that hope alone can help patients scale impediments that sometimes impede treatment, and that hope itself is a medical intervention. So if this is true, then we ought to be able to make some hypotheses and make some measurements. For instance, we could conceivably go back and measure endorphin levels in patients, whether they'd received the actual surgical treatment or not. We could answer whether an endorphin high can last for 2 years. It's an interesting question. It doesn't seem like it would, We understand under which circumstances the endorphins get released. It's from that hope and expectation. But can that hope and expectation really carry on for 2 years? Well, if we could measure endorphin levels in the blood, we might have an answer. We would want to know what the limits are to the endorphin effect, right? So this study had a 2-year post-operative assessment period, which seems like a long time. Does it go on beyond that? And what's going on in that knee in the meantime if he didn't get the treatment? Is there some adjustment in the knee that has a long-term carry forward? Who knows? So there are a lot of unanswered questions, but it looks as if when we take into account Bruce Cohen's work and now Benedetti's work, that this might be something that we want to consider as a long-term medical solution to key kinds of problems. We just don't know yet. We do know a bit more about placebos and how they work. There is no single placebo effect, for instance. Different conditions have different placebo responses. The expectation of future outcomes is a principal mechanism, and that operates when the anticipation is positive. But we also know that there's a certain amount of learning with respect to placebo phenomena, and we can probe this experimentally, and these are data that are reported in the book that Benedetti wrote. When a positive benefit is associated with a placebo, and the placebo is given in the form of a pill, that the pill might need to be a certain size or color in order to exert its placebo effect. In other words, if you are giving a pill to act as a placebo, a small white pill might not be as effective as a medium-sized yellow pill, it turns out. And that's interesting. Not that it is determinative, but it's just interesting that there would be these elements to the placebo effect. It's also the case that some personality traits are associated with responsiveness to placebos. In other words, people with particular personality traits, highly suggestible personality, are more likely to have a positive reaction to a placebo, which actually sounds not untoward. That makes a certain amount of sense. There may be a genetic component to the placebo effect, and indeed, if high levels of suggestibility is a genetic phenomenon, as many of us suspect it is, then that could be one reason why the placebo effect takes place. We know, furthermore, that serotonin is involved in both suggestibility and thus possibly the genetic underpinning of the placebo effect. So that too could be probed. Oops. And we also know that placebo effects may be present in animals. We can do similar experiments, similar to the RA and V experiments in animals with adjustments for the fact that they're animals. There's a drug, scopolamine, that can be used as sort of a check on the placebo effect in animals. And so those are experiments that are taking place right now. In terms of the current status of research on placebos, we certainly know that analgesia or pain killing or pain suppression is the most common type of experimental setup for placebo studies. And as I mentioned earlier, one of the reasons for that is that we know the mechanism for analgesia in the nervous system, and so it's easier to perform studies based on pain suppression simply because we know how it works and therefore how to interfere with it and adjust the experimental presentation more easily. There are a whole bunch of factors that can potentially influence the magnitude of the placebo effect. We looked at some of them on the previous slide, but there are many more that also might be considered. We know that if you slip an actual painkiller into a treatment, but you don't tell the patient that they're getting a painkiller, that the pain-killing activity is less effective. So there's like a negative placebo effect when the administration takes place secretly. We know that some analgesia is, as we've discussed a lot, is mediated through the release of endogenous opioids, but we also know that there are some other receptors and other chemicals that can have similar activity. So there's some cannabinoids that the receptors in our systems, and these are not reversed by naloxone like the ones we've been talking about are, and so that opens up some additional avenues of investigation that are currently taking place. There are other systems entirely that might be investigated. There are placebo analgesia studies that relate to the dopamine reward system and the release of dopamine in the brain. And we also have situations in which the usual mechanisms for pain relief and pain suppression may or may not be operative. In these cases, people with Alzheimer's come to mind because they lack prefrontal control and they don't show a placebo pain response, at least in the same way that normal people do. So there's just this morass of stuff we can look at, many questions that we can answer, lots of different receptors we could look at, lots of different systems we could probe to more fully understand the nature of the placebo effect in humans. And here is your study guide for Module 8.